Hello students, how are you all? Hope that you all are fine as well as healthy. We are learning biology, 12th science biology and chapter number 5, the principles of inheritance and variation. So in this chapter number 5, it is a chapter of genetics. Okay, so the first part or the first portion of this chapter is completed, and in last video lecture we have talked about the some basic and the important information about the codominancy. Okay, and some remaining aspects were also there, some remaining aspects like the about multiple alleles. Okay, then dominance is not a autonomous character, and then one paragraph is there for the capital B as well as small b. Okay, so before starting today's video lecture, we are going to revise the last video lectures at a glance. Okay, so if we talk about the revision of last video lectures, so we can say that Mendel did what? Mendel did monohybrid cross. Okay, Mendel did what? Mendel did monohybrid cross for what? For understanding of inheritance of one gene. For understanding of inheritance of one gene. So we can say that what is done for the understanding of inheritance of one gene? Mendel did what? Mendel did monohybrid cross. So we can say that monohybrid cross was done for the understanding of inheritance of what? Inheritance of one gene. And if we talk about the result, if we talk about the result of which generation? Result of F2 generation. If we talk about the result of which generation? If we talk about the result of F2 generation for what? For the monohybrid cross, then two types of results are there. The first one, we can say that the genotypic ratio. Okay, so the first one, we can say that genotypic ratio. And what about the second one? Second one, we can say that phenotypic ratio. Okay, second one, we can say that phenotypic ratio. So you will remember the genotypic ratio will be 1:2:1. Okay, 1:2:1 means 1 capital T is to 2 capital T small t is to 1 small t means 25, 50, 25. Okay, means 1/4, 1/2 and 1/4. What about the phenotypic ratio? It is the 3:1 means 3 tall and 1 dwarf plants are there. And then 75% is to 25% means 3/4 is to 1/4. Okay, it's all about the monohybrid cross. Now if we talk about what? Now if we talk about the incomplete dominancy. Okay, if we talk about the incomplete dominancy, in incomplete dominancy we can say that what about the result found in the F2 generation? In incomplete dominancy we can say that what about the result found in which generation? Result found in F2 generation will be different. Okay, in the case of incomplete dominancy, the result found in F2 generation is different, means we can say that different comparatively the monohybrid cross. Okay, we can say that in incomplete dominancy both genotypic ratio and phenotypic ratio, both genotypic ratio and phenotypic ratio will be same. Okay, so both genotypic and phenotypic ratio will be 1:2:1. Okay, 1:2:1. So it's about the incomplete dominancy. Now what about the codominancy? Okay, so if we write here, what about the codominancy? In the case of codominancy, codominancy is nothing but we can say that in the generations, in the filial generations, the both characters of parents are observed. Okay, the characters of both parents are observed in the case of codominancy. And what is the best example for the understanding of codominancy? You know that the ABO blood group. Okay, ABO blood group found in humans. ABO blood group of humans is the best example of codominancy. And then one table is also important. Okay, we are again going to write this table. We can say that allele from the parent one, here allele 1, okay, from parent 1, and here we can say that allele number 2 from parent 2. One allele is maternal and second allele will be the paternal. Okay, here we can say that the genotype, here we can say that the genotype, and at last we can say that the phenotype. And you know that the phenotype means what? Phenotype means nothing. Phenotype means one type of blood group. Okay, phenotype means what? Means one type of blood group. Okay, so you know that the total six possibilities are there. First one here IA with IA, here IA with IB, third one IA with small I, fourth one IB with IA, IB with IB, fifth one IB with small I with IA, small I with IB, small I with small I. Okay, so these are the all possibilities for the alleles from the both parents, mother as well as father. Okay, you know the why only two alleles are there? Because humans are the diploid type of organisms. We are 2N, means haploid type, so diploid type of organisms. Okay, so genotype, here we can say that IA, yes or no? It is the homozygous pair. Here we can say that IB. It is what? It is a heterozygous type of genotype. Here IA and small I. It is also heterozygous type of genotype. Here we can say that IB is homozygous type of genotype. Here IB and small I, it is one type of heterozygous type of genotype. Here small I and small I, it is one type of homozygous type of genotype. Okay, so total how many? Total 6 possibilities, okay, are there. Now if we are going to write the phenotype, here blood group A, yes or no? Blood group AB. Remember, blood group AB having the always heterozygous type of genotype. Here blood group A, but see here homozygous A means this blood group A having the homozygous type of genotype, while that blood group A having the heterozygous type of genotype. Here blood group B, this blood group B having the homo type of genotype, while this blood group B having the heterozygous type of genotype. Blood group O having the always homozygous type of genotype, means small I and small I. Okay, so that's all. And now if we talk about the next concept, what about the multiple alleles? Okay, you know the concept of what? Concept of multiple alleles. See, normally we can say that the one gene or the one factor having the how many? One factor having normally 2 alleles, but in some case we can say that this one gene having what? One gene having the more than 2 alleles. Okay, one gene having more than 2 alleles, and this condition is known as what? And this condition is known as the multiple allele. Okay, what are the examples of multiple alleles? We can say that if we talk about the examples of multiple alleles, then first one is ABO blood group. ABO blood group found in humans. ABO blood group found in humans is the example of multiple allele. Why? Because we can say that, you know, the I gene is there. Okay, so this I gene having the how many? 3 different type of alleles. Okay, IA, IB and small I. And second example of multiple allele is what? Is the coat color in rabbit. Okay, what about the second example? Second example will be the coat color found in rabbit. So this coat color means one gene having the how many? You can say that 4 different type of alleles. Okay, it is not included in the syllabus of your state board. So 4 alleles, like you can say that first one is albino, second one is agouti, albino agouti, then chinchilla, and fourth one is Himalayan. Fourth one is Himalayan. Okay, so these all are the four different type of alleles. Okay, so you will remember this and see the third paragraph is there. Okay, on the left hand side page of your textbook, that we can say that dominancy is not what? Dominancy is not a autonomous type of character. Dominancy is not a autonomous type of character. So now we are going to understand this sentence. Okay, what is the sentence? Sentence is dominancy is not one type of autonomous character. Dominancy is not one type of what? Dominancy is not one type of autonomous character. Okay, see here we can say that, see, we are talking about one gene. We are talking about the one gene which is responsible for what? Which is responsible for the production of starch. Which is responsible for what? Production of what? Production of starch in Pisum sativum, means in pea plants. Okay, in Pisum sativum means pea plant. See, we are talking about what? We are talking about the one gene, okay, which is responsible for what? Which is responsible for the production of what? Production of starch. See, when starch will be produced, you know that the starch is one type of polysaccharide and which is made up of the monomers like the glucose. So we can say that starch is one type of last product of the process photosynthesis. Okay, so production of starch in pea plants will be controlled by one gene. If we talk about the alleles for this gene, so capital B is the dominant allele, while small b is what? Small b is the recessive type of allele. Okay, you will remember capital B will be dominant and small b will be the recessive. Capital B will be dominant type of allele and small b will be the recessive type of allele. So here if we talk about the some possibilities, option number 1, if the capital B is there, means homozygous type of genotype. Then if the capital B is there, then see, capital B is what? Capital B is one type of what? It is one type of genotype, and we can say that because of this genotype, starch production is found. Okay, starch production is found, or we can say that high amount of or higher starch production is found. Now see, because of starch production, because of maximum starch production, the storage of starch production is there, yes or no? If maximum starch will be produced, it means maximum starch must be stored. That's why we can say that this capital B is responsible for the round shape of what? Round shape of the seed. Round shape of what? Round shape of the seed. Got it? Yes or no? See, this gene is responsible for that character, but will affect also the different type of character, will affect on the other character also. So it is option number 1. Now if we talk about the option number 2, what about the small b? Okay, see here, small b means what? It is the homozygous recessive, yes or no? That first option is the homozygous dominant, and that one is what? That one is the homozygous recessive. It means we can say that the no or the lesser starch production is there. No or lesser starch production is there. It means we can say that wrinkle shaped. Okay, we can say that wrinkle shape of the seed. So the shape of the seed will be what? The shape of the seed will be wrinkled. Round એટલે ગોળ, wrinkle मतलब ખરબચડું. Okay, round and wrinkle. Now see, if we talk about the third option, the third option is what? Third option is capital B and small b. See, it is one type of heterozygous type of condition, and in this condition always the law of dominance is applicable. Okay, see, law of dominance is only applicable in the heterozygous condition, because in heterozygous condition one dominant allele is there and second one is the recessive allele is there. Okay, but here what is going to happen? Here we can say that less starch production is found, okay, compared to this. Compared to capital B lesser, and compared to small b, higher cell production is found. But see, we can say that the shape will be what? The shape will not be round. The shape will be intermediate. Okay, shape will be intermediate. It means if the capital B is 100% dominant, then Y shape will be intermediate. The shape must be the rounded. That's why dominance is not what? Dominance is not autonomous character. Okay, so it's all about that paragraph, okay, given in your textbook. Okay, now we are going to talk about some examples. Okay, some examples which are connected, some examples which are connected with the question of the codominancy, but I will call it the examples of blood group, and these examples are very much important according to view of the MCQs as well as 2 or 3 mark. Okay, question of 2 or 3 marks. Okay, before starting these all examples, we are going to write one table which is useful for the all examples connected with the blood groups. Okay, so see here we can say that if we talk about the ABO blood group in human, if we talk about the ABO blood group, in human, so see now I am writing the different 4 types of blood group. First one, second one, number 3 and number 4. Blood group A, here we can say that blood group B, blood group O, not blood group C. Okay, ઘણે લખી નાખે A, B and C, એવું ન હોય ભાઈ. Blood group AB, and last one is blood group O. Okay, see these all are the blood group, means we can say that these all are one type of phenotype characters. Now we are writing the genotype. For this 2 options are there, IA, which is homozygous type of genotype. Second option is IA small I, which is heterozygous type of genotype. For blood group B, also 2 options are there, IB. It is what? It is one type of homozygous type of genotype. IB small I, it is what? It is one type of heterozygous type of genotype. What about the blood group AB? We can say that IA, IB, it is always heterozygous type of genotype. You will remember blood group AB having the both different type of alleles, so heterozygous type of condition is there, and the codominancy is only applicable here. Okay, see here in this case law of dominance is applicable, yes or no? One is dominant, other one is recessive. Here again law of dominance is applicable. In homozygotic condition there is no law. Okay, none type of law will be applicable. And here we can say that also homozygous type of condition. Okay, so small I and small I. You will write this for 5 times. Okay, it is extra homework of today. You will write this for 5 times. Okay, before starting or before writing or before learning the examples of blood group, you are going to write this. Okay, તમને એક્ઝામમાં પૂછાય, હવે એક્ઝામ્પલ તમારા સાઈડમાં લખી નાખવું. બહુ પ્રેક્ટિસ કરશો ને તો આ લખવું જ નહીં પડે, તમારા મગજમાં છપાઈ જશે. ઓકે, there are total 3 or 4 types of methods for the examples connected with blood groups. 3 થી 4 પ્રકારના blood groupના દાખલાઓ તમે તેને કહી શકો, examples પૂછાય છે. We are going with the first method. Okay, method number 1. See here, method number 1. Method number 1, example number 1. Okay, see if the phenotype of mother is IA small I and sorry, not phenotype but the genotype. Okay, if the genotype of mother is Ia small I and genotype of father is IAIB, then what are the possible, say I am using what, phenotype. Phenotypes means, in other words, possible blood groups. What are the possible blood groups found in their progeny, means their children's. Okay, see, genotypes are given. It is method number 1. Means we can say that, see here, mother having what, mother having genotype like IAN small I, so the phenotype of mother will be blood group A. And if we talk about the father having genotype of IAN IB, so blood group will be what, so blood group will be AB. Okay, now see, we will use the same method of monohybrid cross. Means here, if we talk about the P generation, we can say that mother cross with father, means we can say that here IA small I cross IA IB. Okay, you are going to use this type of sign for male and female. Okay, now if we talk about the gametes, see, both are homo or heterozygous, both are heterozygous, so heterozygous will produce the different type of gametes but in the same number. So here we can say that 50% IA, 50% small r, here 50% IA, yes, and 50% IB. Okay, so now with the help of what, now with the help of Punnett square, with the help of what, with the help of Punnett square, means you can say that using of what, means we can say that using of the Punnett square. Okay, so it is Punnett square here, IA small I female gametes, here IA IB means one type of male gamete. Okay, so see, the first genotype, option number 1 IA, option number 2 IAIB, option number 3 IS small I, option number 4 IB and small I. Okay, IA small I, IAIB and IB and small I. So we can say that if we talk about the result, then 1/4 IA, 1/4 IAIB, 1/4 IA small I, 1/4 IB small I. Okay, means we can see these all are the what, these all are the genotype. What about the phenotype? 25% blood group A, 25% blood group AB, 25% blood group A, 25% blood group B. See, total 3 types of blood groups are there. The first type is what? First type is blood group A, second type is blood group B, and third type is blood group AB, and third type is blood group AB. It means we can say that in result, okay, we can say that one blood group AB is to 2 blood group A is to 1 blood group B. Means we can say that 25% possibilities of blood group AB, 50% possibilities of blood group A, and 25% possibilities of blood group B. So it will be what? It will be the answer. Okay, see, you are getting what? You are getting 4 different type of genotype, but see, it is blood group A and this one is also blood group A. That's why the blood group A will be what? The blood group A will be 50%. So it is the method number 1. Okay, and remaining 2 methods are also there, method number 2 and 3 and 4, but we are not going to talk about this all remaining method in this video lecture, but I am giving you some examples. Okay, and you are going to write these examples in your homework. So I am writing here genotype of father, genotype of mother, and what will be the genotype of their progeny. Here IS small I, IB small I. What about their progeny? Blood group. You are going to find the blood group of their progeny. Here we can say that IA, IS small I. Second example, third one is small I, IA, IB. Okay, third one, fourth one is IA, IB, and IB, IA. It is the fourth one. Fifth one will be the IA, IB, and IB small I. So total 5 examples are there, and you are going to do this all example with the help of this same method. Okay. So in next video lecture we will talk about the method number 2 and method number 3. Okay. In your exercise, the question number 12 is given. Okay. You are going to try this. Okay. If you are not getting it, then it's okay. We are going to solve this question number 12 in the method number 3. Okay, so first method of this all examples is completed. Today's lecture is 100% completed. I hope that you are getting this concept, this information, this content very much easily. Thank you very much.